Hello and welcome to the Microbiome Medics podcast. I'm Dr. Siobhan McCormack.
And I'm Dr. Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science. We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients.
Hello, welcome to another episode of the Microbiome Medics podcast. I'm Dr. Sheena Fraser. Today, I'm joined by Professor Tim Spector. Tim is a professor of genetic epidemiology at King's College London. He's also co-founder of Zoe. He's an honorary consultant physician at Guy's and St. Thomas's. And he is one of the pioneers of microbiome research and microbiome development in this country. So I'm really delighted to have him here. Really exciting to talk to you finally, Tim. Our listeners would be really interested to hear about your career, about your work, about Zoe. So hi, how are you?
I'm great. I don't know how we're going to cover it all, but we'll try.
It's going to be slightly tricky, I think, but we'll get going anyway. So Tim, let's start way back. Take me back to your initial studies, because you started off as a genetic epidemiologist with an interest in twins, and you've made it from the sort of twin area of study right through to microbiomes. So tell me, how did that happen? Tell me about your early research.
I started after I'd finished my thesis, which was on hormones and arthritis. I decided to change tack and start this UK twin registry. That was back in 1993, because I just thought twins were cool. I studied them a bit in epidemiology, and this, nature nurture idea was super exciting at the time because we had no clue about the genetic component of most diseases then. And we weren't able to use lab methods really to do much. It was pretty crude. So that's when the sort of just looking at correlations between twins was really exciting. And we made lots of discoveries about things that people thought weren't very genetic, like back pain or hemorrhoids, ended up being more genetic than things like breast cancer, which people had always assumed was much more genetic. So we changed a lot of perceptions and moved things, but and twins were really just a tool rather than I was studying twins. So that allowed me, with my restless nature, to study hundreds of different diseases. And so I moved from being a specialist rheumatologist with an interest just in the menopause and arthritis to suddenly studying any number of diseases, because I realized that we had the toolkit to do it better than anyone else, and could be completely cross-disciplinary. So doing everything from psychology to dementia to sexuality to political views on Brexit to, cytokine levels in the blood, or, we could do anything. It was, and people were all often a bit shocked. They said, what are you publishing, and therefore, because in academia, everyone wants everyone else to be pigeonholed, so they know exactly who they are. But that gave me this broad idea, but then, having done some of the first GWAS studies in about 2003, which is genome-wide association studies, where you find this was the very first bit of the genetic revolution, really, that we were using it. It became obvious that genes weren't the answer for common diseases. They were really good for rare diseases, but there were thousands of them. So something like obesity had at least 1,000 different genetic subvariants and things. So that, I thought the practicality of that was starting to get harder and harder. And so I started looking at other things. And one way was to look at how identical twins who were brought up together, why do they get different diseases if they've got the same genes? And for these conditions that are like 50% genetic. And I did all sorts of studies, and I was always looking for some new biomarker. So I would go around to these conferences looking for new talks. And then in about 2009, I first heard of the microbiome, right? In the US, who were way ahead of us in the UK. And you're talking about Helicobacter, if people remember Helicobacter, how that was that microbe that found to cause ulcers, that once you'd eradicated it, caused other problems in the body. And it was naturally sort of meant to be there in humans. And so that was this whole new concept that I thought really exciting. And so we desperately tried to work with a US group to get microbiome tested in the twins, because there was no one doing it in the UK. And eventually we've got this group with Cornell that were doing it. And we did about 400 twins in the end. And this is around 2011 when we actually sort of got that up and running. And then, bingo, it turned out that, there was hardly any genetic component to it. And identical twins had very different gut microbes. And so I said, wow, this is the first thing I found in years of studying twins that's different. This must be significant and must be why we all get different diseases.
And way back then, what was the microbiome analysis like in comparison to now?
Well, we used 16S analysis then. So this was the first lab one of the first labs in the US that were doing 16S analysis for people. I don't know how much the listeners know about this, but before that, you could do a PCR test and get, say, 10 microbes, and you would get a little marker for each of those 10 microbes, and you would run them. So that was what was done in quite a few places. Say Nestle or Danone would do that stuff for their yogurts, and that was what was used. Then the 16S thing was really quite revolutionary. It came in around this time, perhaps to be used about 2009, which just measured one gene in every microbe has. It's like the HLA system of microbes, and then guessing how that gene differed between them, you could make a pretty good guess about what the microbes are. And that system is still used today. So, interesting, most gut testing systems in the UK still use that old system. Okay. So it's not as outmoded as you might think from the date, given that it was 14 years ago. So we got decent results, and we've repeated them with our new metagenomics, where you measure every gene in every microbe, and they get the same results. So, I think for showing broad patterns, the old ways were still fine. They just didn't show the detail or the species or the strains. Yeah. And so, but that led me really on this path of, I must spend the rest of my career, not just showing things are genetic, although that was really good for my CV and my H index, but move into this area because of its massive impact as the link between food and disease. And I think that's what, and it so happened that I had my own personal story, which just happened to coincide at the same time, which selfishly motivated me to find out much more about it as well. So that was an additional driver. I had this mini stroke in 2011 that left me trying to work out what I could do lifestyle-wise for myself, and that included changing my diet. So those two things came together.
And was it around that time that you wrote The Diet Myth, or was that just after?
I was just finishing another book called Identically Different, which was about epigenetics, which up to that point I'd been really excited about, because I thought, well, maybe little chemicals or the way you eat or vitamins can change your genes, and they can, but not that much. And so it was fascinating theoretically, but wasn't very useful practically. But in this book, there's a page in there on the microbiome, which I think is about the first sort of popular book on that subject. I was sort of guessing that this is going to be big, so in the final proof stage, I stuck in a page on it. And but obviously, you write on books a long time before they come out, and so I then started on my next book at that point.
Yeah, of course.
Got very excited, and that was really the germ for the diet myth as well. So everything was happening. I was doing the research, I was writing a book on it, and I've always found writing a book is the best way to learn a subject.
Yeah.
Because I'm somewhat unusual medic in that I've changed specialities quite a few times, and you either write a thesis or you write a book. Or you go on some 2-year sabbatical. So that's what I did. I did that with epigenetics when I switched, and I did it with the microbiome. And learning the literature behind a subject, even for the public, I think is a really, really helpful. Gives you this really big overview that actually most academics don't have, because they've gone into it from a lab position, and worked backwards.
No, I can relate to that. I spent over 3 years writing a course on the gut microbiome with my colleague, Dr. Siobhan McCormack. And once you do a really big deep dive, once you start writing about a subject in that kind of depth, you really have to know your stuff. And it was a big eye opener for us, and we learned a lot in the process. So it's a great thing to do. I actually read that diet myth of yours back in 2018, and I have to say, I'd only just heard about the microbiome back in 2018, and I read your book with real interest, because I've always had a real interest in diet and food. I grew up with parents who were very interested in organic food, organic gardening. My dad did research into pesticide. I was watching documentaries on intensive farming when I was a kid. So diet's always been something I've been very, very interested in. So your book was it was a great book for me back in 2018. So I enjoyed it.
Yeah. Not many people read it back then, so you were one of the few, so ahead of the curve.
Yeah, slightly ahead of the curve. Not as ahead of the curve as you are. Obviously, I was slightly late to the microbiome science, and compared to you guys. But it was a big eye opener, and I suddenly thought, gosh, this microbiome is something I need to know a lot more about, and that's when I started madly researching everything I could on it. So we're going to come back to your health issues and what you did in terms of your diet. I think that's something that I'd like to cover sort of later on in this chat. But let's go back. So you started looking at microbiome. When did the British Gut Project start?
Pretty much just after that. So through Ruth Lay in Cornell, I met a guy called Rob Knight, who was in Colorado, who was setting up the American Gut Project. And I went there because I had a sort of similar idea of doing something in the UK, which was a sort of crowd-funded project that primarily for research, people took a stool sample, and then we got this picture of what was going on. And it turned out it was a lot easier just, rather than setting up my own lab, to work with him and have a British Gut Project that had identical protocol to the American Gut Project. And so we combined forces and worked together for several years, and produced some interesting papers and insights, really, particularly comparing the two populations and things. And it was this idea that suddenly you could do this remotely, didn't have to come to hospital. The whole concept of sending poo in the post was quite novel. There was a lot of ethics and things to sort out. People were a bit squeamish, particularly in the UK. But we solved those problems, and we got some interesting early insights into that before the system broke down a bit just because it was hard to keep it funded and the logistics, etc. But and then that sort of morphed into. I then tried briefly to start a little company myself, doing this at King's, which was called Map My Gut, and then my colleagues from Zoe came along and said, "Well, stop all that silly stuff. We can do this on a proper scale, not just academics playing. We can do this really well," and move to the next stage. So this was all 16S until this point. So it was the sequencing that changed
and
it.
If we go back to the British Gut Project for a wee minute, how many samples did you get with the British Gut Project?
I think we got about 5,000, something like that.
Okay.
Possibly about 8,000 it was, yeah.
And what were your main findings? How did our British guts compare to the American guts?
Well, we were better, but we were terrible compared to everyone else. So it was like it's coming, second bottom in class. And because British gut ended up taking a lot of other European samples as well. And generally, everyone was healthier than us, but we could rely on the Americans to have even less healthy guts. And that's still true today when, looking at the Zoe data, interestingly.
Yeah.
So we're having a bit of a dilemma about scores. Do you have a score that's unique for that country, or do you have a score that's, should be global? Mm-hmm. Where are the limits set? So that was the first study that really compared different countries, and I think got people excited. This idea of this as a sort of home test kit that could be done cheaply, because the lab that in the US have managed to streamline things to get it down. Because privately you'd still be paying £500 for this at the time. So I think we got it down to about £70. They were interesting pioneering times when we didn't really, and it was still crude. We couldn't tell exactly what the species were. We didn't know. There was lots of misclassification, and we used this broad definition of diversity.
Yeah, I was going to say, how did you define what was a relatively healthy gut back then? What did you use?
Pretty much the same way as being done now. Hasn't changed. It's there's this sort of alpha diversity that's that says within your gut how different are the different species and microbes. How much variation there is, which is so that has been the core of all microbiome research, really, whether it's in rats or in humans. So that's still true today. It's still the gold standard method, and even going to metagenomics, where you've got much more data, generally the same principles apply. Some people use richness, some people use gene counts, but they're all sort of highly correlated with this approach, which I think worked pretty well for 20 years.
Okay.
I think we can discuss how it's time for a change now, because it doesn't work very well in trials. It doesn't work very well when you interventions.
Yeah.
And there are problems with it.
And tell me about metagenomics. Our listeners may not have heard that term before. Do you want to explain it?
Yeah, another word. It's a bit of a confusing word, really, because some people, particularly some companies trying to sell their online products, call 16S metagenomics, because you're looking at multiple things, the genomes of multiple different creatures. Because, but what it generally means is shotgun sequencing of everything that you can find DNA on in your stool sample. So you get the DNA, you break it apart with these beads and this sort of preparation step, and then you break it into these tiny fragments, and then you're trying to identify every gene and every microbe, and then like a giant jigsaw puzzle, put it back together again. And I think that's what we should all be using now, because it allows you to get to the species level, it allows you to get the strain level. You correctly identify microbes that we were calling something else in the past, and we've moved away from this. You might remember we used to worry about firmacute Bacteroides ratios and all this sort of stuff. That was just because we didn't really have it. We were grasping at straws then. That's all pretty useless.
Yeah.
And because we're all so different, we've all got very different patterns, and our microbiomes are only sort of around 20% similar. And strain sharing really is quite unusual, and if you're not in the same family. So, so that's metagenomics, and it can be, if you take it to its extreme, you can start within metagenomics to measure viruses and fungi as well. Amazing,
and of course that's allowed us to recognize more species and to discover more species. So of course the number of species that you're able to find now is expanding all the time, isn't it?
It is. We think we're about identified about 80%.
Right.
Now it doesn't mean we know what they are. We've just given them a number, but that literally went from 50% to 80% in 2 years when we just updated the software, which is this sort of massive database that can then be applied to any sample. So it's very cool, and we wouldn't have been able to do this without the gene revolution, all the human genetic stuff, the technical ways of getting relatively cheap sequencing, and we couldn't do it without massive computing power and cloud computing, which is what you need to do this stuff. So, I mean, the only problem is it's taken it away from the individual researcher. You now need to, have these massive facilities handling sort of 400, sequences a day to make it cost effective.
Yeah, I was going to say that can't be very cheap either for the average scientist who's wanting to study microbiomes.
No, who wants to do 10 samples? No, it's like horrendous. So yeah, and in the UK they never invested in any large facility to help scientists at all, whereas in other countries they did. So that's one of the reasons why the UK is behind the rest of the world in microbiome research. Is everything was going into genetics, but not really to the microbiome.
Yeah, actually, I was going to talk to you about where we're at with microbiome research. This might be a nice time to do it. I was talking to Professor Karen Scott from the Rowett Institute, and she was talking about how the research that they want to do is not necessarily the research that they get to do, because, of course, it relies on funding, and whoever funds your research is very much going to want to have a say in what you're researching. And of course, if that funding comes from big food, or if that funding comes from big pharma, then they all have their agendas, and they want research to be traveling in a certain way so that there is a commercial product at the end of it. And I think that that seems to hold up research a lot. I was chatting to two separate PhD students who are about to start PhDs in science. And I said, well, what are you going to study? What are you interested in? And they both told me it doesn't matter what we're interested in. We have to study basically what the university tells us to study. And that's where the funding comes from. So what do you think about that in the UK at the moment?
No, it's pretty bleak. It's pretty sad that, most of the ones I see, are related to food companies or, testing some new yogurt or something, which isn't really going to, help mankind that much. There are exceptions, and there are some good places, but, I think a lot of researchers have been forced to go to big food companies for funding, or probiotics companies, just because there aren't any significant other big grants out there for them, because there's no one championing microbiome at the central, in the resource centres, in government, in MRC, in BBSRC, the ones that give all the really big grants. And so that having to do very commercial deals, not because they want to, but, if they want to continue as an academic, that's what they have to do. So, yeah, it is a bit bleak. And of course, Biobank, where we've pumped millions and millions of our funds, was obsessed with genetics and MRI scans and biomarkers, and still hasn't really done the microbiome, although, for at least 10 years I've been telling them they should do. But that's just because those people are not trained in it, and most of my genetics colleagues are pretty dismissive of microbiome research. They see it as a sort of inferior branch. And, always distrustful of new areas. I think they think it's, a passing fad, or, overhyped for commercial reasons, or whatever excuse they use. So, yeah. Whereas, you go to other countries, Germany, Netherlands, France, the US, Denmark, it's pretty big.
Yeah, they're pushing forward much more, aren't they? I was chatting to a guy in Amsterdam, and he was saying that they're building a huge fecal transplant facility. They're really pushing microbiome. A GP I spoke to over there was saying that, GPs all know about the importance of the gut microbiome over there, and they understand the importance of nutrition, diet, lifestyle in enhancing your gut microbiome. But that's not the case in the UK. And I think that's very sad. I mean, obviously I'm biased. This is an area that I'm really interested in, but I feel like we're missing a real trick at the moment, and I think it's very sad when the reason why we're not up to date in this field is because it's political, it's financial, it's due to the way that funding happens in this country via big organizations, big multinationals, and I don't know how we go about fixing that situation.
Yeah, I mean, exactly. So it's the fact that, a lot of the microbiome places around the country do get money from the food industry sort of means that, oh, well, the big institute, the big fund, the independent funders say, oh, well, they don't need the money. So it's a bit of a vicious circle as well. But I think we've had a rather blinkered view, I think, of research in this country that's been dominated by big food. No decent study has ever been done on ultra-processed food in this country. I mean, just think of that.
It's incredible, isn't it?
And yet, we're spending hundreds of millions on rare disease genetics, which is admirable, but it's not helping our population. We spend hundreds of millions on Biobank, which is mainly being used by the Chinese now for their publications because they have free access to it. So I do think we need think about nutrition. Nutrition generally is massively underfunded, and the science of microbiome, which I think underpins a lot of it and gives it perhaps more credibility than it, and higher impact profile than it had in the past, meaning you can get papers now into Nature and things on food that you would never have done 10 years ago. So, yeah. Anyway, we can go on whinging about it, but I don't think either of us will be able to change it. But it's good for people to know,
exactly.
Anyone listening, and if they are, in a position to whisper in someone's ear, we just need to be championing gut health, gut microbiome, get it part of medical training, get it like in other countries and learn from other countries, realize just admitting we're so far behind, I think would be helpful.
Yeah, it's a good way to rebel. against Big Pharma if you're feeling rebellious. Okay. And actually, what is interesting, Tim, is given the culture and the way things are set up for microbiome scientists in this country, what you've achieved with Zoe is all the more miraculous. So I'd like you to talk us through, Zoe, how that came about. I mean, just even how you got the funding to create Zoe, I think is really impressive. So talk us through it.
Yeah, so it was 2017. I was giving a talk about the dark myth, and a couple of guys were listening in the audience. George and Jonathan, my co-founders, and they basically came up to me and said, "We'd like to chat to you." And I thought I'd never hear them. Then they persisted, and finally, I met them at St Thomas's Hospital, and they said, "We'd love to do. We've got an idea of a startup based on personalized nutrition plus the gut microbiome. We think there's this new stuff about CGMs, glucose monitors. Mix that with the microbes. We think we've got something." And we chatted for about an hour. I told them that we need to do a hell of a lot of research before you've got anything that you could, sell, because I'm not interested in just a marketing-led product, which most of them are and still are today, because it's much easier to raise money that way. So I thought I'd never see them again, but they came back with about 3 million quid from a series of angels and people they knew who believed in them, and that some of them read my book, and they believed in this dream that you could do something. So that allowed us to do this massive study, the PREDICT study, which we did in, I think, in just over a year, which would have taken 5 years academically, and would have cost probably at least £5 million. So we'd never have done it, essentially. And this massive study of 1,000 twins, people who don't know it, was quite revolutionary. No one had ever done a food intervention study where you measure everything sort of before and after, and looking at their microbiome, etc. So it was really cool, and that got the whole thing off the ground, because we showed that there was this huge difference the tenfold difference between how people responded to the same test muffin in terms of their blood fat and their blood sugar. And some of that was influenced by their very different gut microbiomes. And we showed that then you put all this together into an algorithm, you can start predicting how people respond to any food. And that was when we knew we had a unique product that, the Zoe team could then make into an app and a program of how you shift people's diets in the right direction. And it's evolved a lot in that time, in that sort of 7 years, and it's still evolving. But the nice thing is we've now got over 250,000 people's gut samples, and they've donated it for science. We can, we've written, 60 odd papers now. Wow. We've got one, in the top journals, the Lancet, Nature Medicine, Nature, Cell, right at the top, which is very unusual for a company to even bother doing that. And but for me and my fellow scientists, it's a dream come true, really. So, who wouldn't like a free database of 250,000 people and their health, nutrition data, and their gut microbiome to play with? So, I mean, the problem is it's so big that you can't just go on your computer and have a look. That's the downside. So anything takes sort of weeks to run on the cloud. So you've got to think of your questions quite carefully. But amazing. But we are getting this massive snapshot of certainly English-speaking world's gut microbiome.
Yeah.
Which is pretty cool.
Talk our listeners through. Not everybody will be really familiar with Zoe and what that actually involves. So talk our listeners first of all through what happens when you enter Zoe. So and what you actually have to do as a participant in Zoe.
Okay, so there's various levels of Zoe. Anyone can sign up for a newsletter, and there's a podcast on nutrition, so everyone can get that for free. If you've got a bit of spare cash, you sign up for a phase, which is the testing program, and then a subscription. And that's really, so the test phase, you get a continuous glucose monitor, you get a postprandial fat test, you get some test cookies, you get a gut microbiome test, and you get an assessment of your diet in that first week. And then you send those results off. Then you get results back that give you individualized scores based on those 3 parameters on how foods are going to affect your health. And so a score, every food you have will have a score out of 100. So a Coca-Cola might have 0. You might find an avocado is 100. And the ones in the middle will vary depending on whether you're a sugar responder or a fat responder, and to some extent what your microbe is doing. And that algorithm is evolving. We published the initial results, and so we used that initially, but that was on 1,000 people. Now we have several 100,000. Obviously, we have the ability to keep changing it. So the idea is that you shift people's views on food. You make them think more carefully before they eat their croissant. You think telling people what time to eat as well. So we're conscious not just what you eat, it's how you eat, it's how fast you eat. It's teaching people to recognize what ultra-processed food is, teaching people really educationally, what is the best food to eat for your gut. We're giving people clues to try and get, the key principles in everyday life so that you get these habit loops. So, there are 6 principles I discuss in the Food for Life cookbook that, eat 30 plants, try 30 different plants a week, eat the rainbow, eat fermented foods, switch from meat to legumes for protein, have regular fermented foods, at least, 3 portions a day, give your gut a rest with time-restricted eating, and ignore calories and think about quality, not, and what's in the UPF food. So those things continuously keep, we remind people on it. And, the results have been very good. So people who do it for more than a few months, these energy levels improve, mood improves, gut symptoms improve. There's some minor weight loss, but that's not the main factor. People just feel a lot better. And that's, in our trials, we saw this really big early effect on the brain, which was really interesting before it affected, constipation, or we saw big shifts in the gut microbiome. So we think that just switching your diet makes your microbes produce, really good brain chemicals that switch it on. So, as you know, medics don't normally ask about energy levels. It's a sort of, oh my God, if a patient comes in talking about that, you feel crestfallen, But I think we learned very early on that that's one of the big things that changes. And if you want to motivate people to continue or switch diet, we should be focusing just as much on the brain as on the rest of the body.
So that's quite interesting. What if somebody is a participant in this and they make some changes to their diet. Obviously, you're measuring the continuous glucose, so you're seeing those highs in glucose with certain foods, and you're able to say, well, those foods might not suit you so well. You might want to concentrate on foods that don't spike your sugars so well. You're seeing those changes in the gut microbiota, and you're able to say, these foods are causing more disruption to your microbiota. These foods might suit your microbiota better, and then people are going down the route of changing their diet. How quickly will they notice the brain changes, the improved energy? Did it also change mood?
Yeah, within a week. Usually,
within a week,
we saw the mood and energy happening. That was, I'd say, one of the first things that we saw and we've seen this in several trials. So we obviously we did the PREDICT study was just an observational one, and then we did something which we called the METHOD study, which came out last year, was a randomized controlled trial against low calorie, low fat advice, and we saw big changes there really pretty early on in the people that were sticking to it. And we've done another study looking at we've got a prebiotic as well. And we get similar effects. And just looking at our fermented food data, we did a citizen science project on that. Again, mood and energy seem to be coming out as the top one in all of these things once you improve your gut. And I think that's a really underrated way of telling people, this is working. Because they often don't, why should I carry on? My blood pressure hasn't changed, or my cholesterol hasn't changed. Well, it's a fairly new concept to get people to think about recording, how their brain's doing. What's your mood? 3 hours after a meal, is it different to what it was on your usual breakfast? I think these are really important switches. And I personally, when I've switched from my carby, low fat diet of sort of 12 years ago, my mood is so much better, and I've got definitely much more energy. And you realize that sometimes eating is bad for your energy levels.
Yeah, I'd agree with that. I used to have bread at lunchtime with my soup every day, and I realized I had a slump 2 hours later. I would always, my energy levels would drop. I would feel tired. My concentration was poorer, and just getting rid of the bread at lunchtime actually improved my energy, made me feel a lot better. So long as I had a soup that contained lots of pulses and grains and, things that kept me going, I actually had more energy in the afternoon with fewer calories. So,
well, it used to make me hungry. I mean, most people know this. And we did one thing we saw was that about 1 in 3 women had a sugar dip after their spike, after a carby breakfast. And that made them hungrier than if they hadn't had that dip. So they would eat another 500 calories that day if they'd had that spike and dip. Yeah. So that's why you reach for the McVitie's at 11 o'clock. That good Scottish company.
Absolutely. Well, and of course you can theorize why that's happening. Obviously, short chain fatty acid production is higher if you eat things that are sort of more complex, more fibrous. And so you've got more feedback from them to your brain. But also if you're fasting in between meals, so if you're not hungry in between meals and you're not snacking on fatty, sugary things, you're not going to get that spike in the unhealthy microbes, presumably from eating the fatty, sugary things. Plus, you're going to enhance that population of microbes that like it when you fast. Presumably, is that what you think's going on?
Well, it's pretty complicated. I mean, I think that's one element of it. But when we did this massive project, we looked at tens of thousands of people, say, doing intermittent fasting. We found about a third of people really didn't cope with it at all well. And so there is a bit of individual differences between us. So what works for you and I might not work for someone else. So I think we have to keep an open mind and realize there might be different ways to optimize your metabolism. It is, even in the timing, it's personalized. And we see this, and not only between individuals, but also at different ages. So around the menopause, women get bigger sugar spikes than they do 10 years before, and might explain why they get hungrier, why they gain weight, or the metabolism is different. And as you get older, the difference between morning metabolism and evening metabolism is sort of goes away as well. So, this idea of we're being told you must eat everything in the mornings, which is pushed by the food companies, is based generally on students, 10 students who are in sort of metabolic chambers and things, under the age of 21. So they never tested oldies like me. And when we look at my data, I don't change in the morning or evening. There's no difference. So lots of things. You can have a sort of general principle, these things are good, but I think everyone needs to explore their own personal milieu, which, what suits them the best. And hopefully just this whole idea of introducing Zoe, even if you don't want to do the full test, should encourage experimentation, and we move away from dogma. So we move away from saying, never go to work without your breakfast, or children will fall apart at school if they haven't had massive amounts of cereal or a midday snack. It's nonsense. Listen to your body. Millions of years of evolution have given us this incredible tool called hunger to tell us when we should be eating.
Yeah, absolutely. And that's what I really like about Zoe. It's this individual approach. And, I'm a GP. I speak to loads and loads of patients all the time about their diet. And there's a big difference in the way people approach eating, they approach their diet, the way they like to eat, how often they like to eat, whether they fast or not. And some people thrive on fasting. Some people thrive on eating more regularly. Just because there are some health benefits of one approach doesn't mean that it suits everybody, like you say. So I like this because I don't like the one size fits all. I don't like the idea that everybody should be taught the same diet. And I think that that's a really, really good approach. And you're able to obviously give patients the evidence. You're able to give them the readings from their body and their individual responses to foods. And I think that that's so exciting for the people that can afford to do your program. I think that that's really, really exciting and can probably make quite a difference to a lot of people. But if you can't afford to do your program, what would your tips be for those individuals that just don't have the ability to do it at the moment, but want some ideas as to how to work out what's good for them and what's not?
Well, they could read my books; they're cheaper. Listen to the Zoe podcast, which is free. Get themselves better educated in this area, and experiment. Have a look in your fridge, see what you've got, work out what write down what you have most days, and whatever it is, set aside a couple of weeks to experiment with it. I would suggest breakfast is the best one to experiment with, and if you have your Special K or your favorite granola or muesli, have that for a couple of days. Have a little notebook and just say a little alarm that goes off and say 3 hours later, how do I feel? Rating your hunger, your mood, your energy level, and then do that for 2 days, swapping it for like full fat yogurt, nuts, seeds, maybe some berries, and do the same. Then do that for 2 days where you don't have breakfast before 11 in the morning, and see how you feel. So I think that's how I would approach this. I think, in general, most people respond badly to carbohydrates. 2/3 of people, I think. So, that's probably the thing to experiment cutting out and seeing. We're talking refined carbs here, not high fiber carbs, and see how you feel. And I think you just with those three things: mood, energy, hunger. You can see if you're on the right track. And if you think it is important, then I would say it is worth then doing Zoe and getting the app and doing it, because now the price of subscription is now down to below it's $9.99. So you think how many subscriptions you have for $9.99 that are not really going to help your health. I think these things, we ought to be spending more on our health than we do, and on food generally. Learning what the good foods are.
Yeah, absolutely. I would also say that you can work out what foods suit you and what don't. I did this years ago. I realized quite early on as a GP that I was, there were certain foods that would really drain my energy. And I realized that a lot of carby things, porridge, for example, wasn't suiting me in the mornings. I would feel starving hungry by 10:30 in the morning. And I had to switch it. I switched it to a homemade muesli that has tons of seeds and nuts in it, but big jumbo oats, organic oats, and whole fat yogurt and fruit, and that seems to keep me going till 2 in the afternoon, no problem at all. So
not everyone does that, but I think but that's interesting. So we've done these studies, and some people notice when they're getting sugar spikes and sugar dips, but a lot of people do not.
Yeah.
So what I'm saying is, give it a try, and you may well be one of those people that you can pick up what's happening inside your body. But other people are getting these big sugar spikes, sugar dips, and they don't realize that that's affecting them, and they're not getting those symptoms. So again, that's the nice thing about doing these these home monitoring tests, or with these apps or tools. It's a bit like your sleep quality, or your heart rate variability, and these things. Some people are good at working out how they're feeling. Others might need these tricks and devices. Yeah. So I
Fantastic.
think, again, there isn't one size that fits all.
That's right. One other observation as a GP, everybody that I diagnose with pre-diabetes or diabetes is having toast or cereal for breakfast, sandwich for lunch, and then loads of pasta, rice, potatoes in the evening. Every single patient, with snacks in between.
But they're having low, and they're calling it low fat and healthy, aren't they?
Yeah. So, it's obvious that we're consuming more carbohydrates than we should. I think if we don't want to develop that metabolic dysfunction, and then I really think that we have to address the amount of carbohydrates that we're consuming, and think of alternatives. Think of other things that we can consume in place of them that aren't going to be so carb rich or so processed carb rich.
But we've got to break the hold of the food industry that's still pushing the low fat message, and low fat, high starchy, ultra processed foods are the savior of the UK population, and it's killing us. And it is just outrageous that there aren't bigger public health campaigns. Where's Public Health England, with big posters saying, a third of the population have pre-diabetes, stop eating white toast. It's like, this is just sugar. We're doing a terrible job.
We are doing a terrible job, but unfortunately, big food is very influential to the government, and I don't think that's going to change unless big food are outside of government agencies, like the Food Standards Agency, for example. Big food sit on that panel. So, how are you going to make objective decisions about the food industry when they're already a part of the discussion?
Well, the Lord's report, which I contributed, has been sent to the government, so we're waiting for their reply. And I think everything we've been discussing here about how to change the system is actually in that. So it depends,
Fantastic.
Do the government have the balls to actually do anything about it this time? It's the key question.
And even if you take it to a community level, what about things like school dinners and hospital food? I mean, hospital food is something that really, really ought to be addressed by now. It really concerns me that the health of people's gut microbiome actually worsens as a result of consuming hospital food when they enter into the hospital environment. And that's going to have a big impact on their response to treatment, the impact from the antibiotics that they may have, the impact from any operation or procedure that they may have in a hospital environment. That could be made substantially worse by the nutrition that they're given in there. What's your thoughts on that?
Well, I agree completely. It's a national disgrace. I mean, my mum was in and out of hospital, the last 5 years, and in 3 different hospitals, and only one of them was it passable. The others were inedible.
Yeah.
They're just slopping up.
That's how my patients describe it: inedible
slops, and baby food as well. So that. It's the worst of all worlds, really. That the people who need that nutrition most get the worst possible food, full of chemicals, and no fibre, no nutrients at all, and just with sweet drinks. And everywhere you go is full of sweetened sodas and vending machines. It's unbelievable. And if you go to other countries, they get really good quality food in their hospitals. I worked in Belgium for a while. They ask you how you want your steak cooked. Could you imagine that, Anna?
Amazing. Amazing.
We just say, it's not worth, it's the budget. It always gets cut first, in schools and hospitals. And I learned recently in Japan, they have a nutritionist at every school.
Japan's school food's incredible.
And they take that incredibly, and they do not let parents contaminate with bringing in junk rubbish.
Yeah.
So they control it. They know how important it is for that child's education. And I think anyone who's a parent should be contacting their local head teacher and saying, do something about the food. I'd love to come in and taste it. I think that would have a huge impact because a lot of them want to do it. And interesting that, like there are a few hospitals that have good food, there are a few schools that have good food, showing it can be done.
Well, the Soil Association Food for Life campaign has been quite influential in school foods, and they have made some really positive changes. So our local school here is a bronze medal holder for Food for Life, which doesn't sound like much, but what they have done is they've got rid of processed meat. They've cut out sugary puddings and muffins and things like that. And they get an organic veg box delivered every week that they have to use in the cooking. They've got to source 70% of their food from the local community, and things need to be cooked on site. So they're not utilizing so much processed food, which is good. But the problem that I see is that, we haven't really paid for chefs or cooks, proper, educated cooks in these environments for a long, long time. And so you're reliant on people who aren't necessarily trained in cooking to carry out the cooking for the school.
And, they're just used to microwaving, aren't they?
Yeah. And so, the complaints I see from my children are that, the food would be good if it was cooked properly. They have a load of oven roasted vegetables, for example, but they're always black and burnt. And, the salad is always chopped up way too early, so it's always dry and, sucky. And so, things become very unappetizing for the kids because of the way that they're produced, which is a real shame, because I think that a lot of work has gone into that campaign to improve school meals, and there's a lot of real benefits to what they're doing, but the way that it's being carried out isn't sufficient, and it's a shame that we're in this situation that it's still not good enough for the kids. So, Tim, let's just go back a little bit. I just want to talk a bit about your findings from Zoe, because you mentioned that you've published 60 papers so far. And we know now through your research from the PREDICT 1 and 2 trials that people are responding differently to foods. And that was your sort of main finding originally, that we can all eat the same food but have a very different microbiome response and glycemic response to that, and that's been absolutely incredible for the nutrition world, for the medical world. This is important knowledge. What else have you discovered? Because I think we would all want to hear what insights you've got. With over 100,000 participants, you must have some really interesting data now.
Yeah, I mean, obviously, that we've talked about different responses. I think we've linked these fat and sugar spikes to inflammation, and inflammation is one of the themes that just keeps emerging as the underlying reason that things are bad for us, whether it's stress or it's food or obesity. So that we're beginning to get some unifying patterns here, that if you have microbes that produce inflammatory chemicals, that's going to be pretty bad for you and your brain, and depression, so it's going, all over the body. I think this is what we're seeing. So obviously avoiding the sugar spikes, the fatty spikes, we know that you get your inflammation levels down. If you produce your gut, you get greater diversity of gut microbes. You get less inflammatory chemicals produced, and your immune system's calmer. So we're seeing a sort of unifying of a number of different ideas coming together, and we're moving away from this diversity score as being the best one. So we've got some data now confirming that taking a ratio of the top 100 microbes that are involved in your health and in food, the ratio of the good and the bad is a much better indicator of your overall health than just how many different ones you have. So we're moving towards a unified concept of what gut health really is and what the sort of optimal gut health should look like, which I think has been a bit of a problem because it's always been a bit circular. So that's going to be quite exciting, because we've got these same bugs that work in the US and the UK. Therefore, we know they're very frequent, and so everyone's got these as little markers of their health. And I think we're now, by interrogating our database, we know how fussy these microbes are. So we found, I don't know if you saw that paper we did on coffee, and the microbe Lawsonobacter only eats coffee. And so that is sort of mind-blowing. You think all these microbes, they're just waiting for you to eat this particular food. You've never had a Jerusalem artichoke, and suddenly, a whole little group get very excited and start multiplying when you do. So that was the first of a number of studies where we're going to start linking the specific foods to specific microbes. At the moment, it's a bit general. We haven't got that fine detail, but now we've got, these 250,000 samples. We can start working out, which. So we say nuts and seeds are good for you, but can we tell what microbes they switch on when you have a bag of nuts, and can we then personalize that to people who don't have very many of those microbes? That's the next phase of what we're going to be able to do, which is quite cool. We're also going to do things like hopefully tell whether you're able to cope with eating lots of meat or not, because microbes break down some of the products in meat to produce these chemicals, TMA and TMAO, that can cause heart disease. And we know that differs. So if we can identify those microbes, we can tell people, you should really limit your red meat, or go ahead, it's not going to cause you heart disease. I think, and similarly with estrogen and soybeans in women, some people can break down soybeans depending on their microbes. So I think we're going to move into this next phase of sort of personalization that's going to be really, really cool and give people these really very specific insights. As well as, morning person, evening person, are you a dipper? Are you?
Yeah, interesting. And the TMAO thing is quite an interesting one, because we link TMAO production to atherosclerosis, or furring up of the arteries. But in the Japanese population, they have high levels of TMAO, but they don't have high levels of atherosclerosis. So how does that work? Are we any closer to understanding that?
Well, I think we'd speculate they've got a very different set of gut microbes, and so they are eliminating it or making sure it doesn't affect the vessel walls. And so I think, in the same way that, drugs have different effects in different populations because the microbes are breaking down the chemicals in those drugs, whether it's paracetamol or antidepressants or warfarin, who knows? I think we have to start thinking very differently about these little differences between people. Stop assuming that everything is always the same. And, often in epidemiology, there have been these differences between Western populations and Asian populations. And soybeans is one example. They're protective against cancer in Asia and either neutral or possibly harmful in the West. Some of that might be the foods are different, but some of it might be the microbes are very different. And
I've often
that's why
wondered
the seaweed eating microbes in Japanese, of course, that mean that they can get many more nutrients from seaweed than we can.
Yeah, so I've often wondered about that, because, of course, in the UK we eat a very broad range of foods now, because we've taken so much influence from all across the world, and we're really interested in expanding our diet to eating Mexican and Asian and Indian cuisines all the time. But I do wonder whether our guts are adapted to that, and should we be eating all of this big variety of foods, or should we stick to what evolutionary we've presumably evolved to eat, and especially on the sort of fermented food side, I do wonder about things like kimchi, because aren't there some microbes in kimchi that are quite different to the microbes in the fermented foods in Europe, for example? So should we be encountering these as Europeans? What's your thoughts?
My thought is we should go for everything. Yeah, and we are at heart omnivores. Humans are incredibly flexible. We can live in totally different environments and have learned to adapt. And in a way, gaining new microbes allows you to adapt to those conditions and eat and digest those foods, whether it's seaweed, it's mushrooms, whether it's meat, it's whale blubber. So I think we shouldn't be frightened of new things. And we did a study which we published a few weeks ago, on difference between vegans, vegetarians, and on omnivores. And although there were broad differences between the groups, and vegans generally were healthier than vegetarians who were healthier than omnivores, within each group there were huge differences. So you could still be right at the top of healthiness by being a meat eater. And actually, there was a suggestion that people who ate a small amount of meat and had all the plants and dairy had the healthiest looking gut. And so I think more is always better. In my view, I don't think there's anything that we shouldn't be eating that's, real food rather than chemical food. But so I'm a big kimchi fan, so perhaps I'm biased.
Excellent. And on the meat front, how much should we be eating? What do your studies say? What's a sort of healthy level of meat eating if you're a meat eater, and what should you not be consuming? How much should you not be consuming?
Well, we don't have the results of our TMAO analysis yet, so I can't really say anything specific from the Zoe studies. The broad epidemiology studies, they tell us the Americans eat twice as much as the Brits in terms of meat.
Wow.
And we eat probably slightly more than average in Europe. Okay. So I'm sure we could all be eating less. I think if it's, processed meats are a different thing. They cause cancer and heart disease. But if we're talking about good quality sort of meats, then in my view, is we don't really know all the answers, and there's probably likely to be big interpersonal differences. But the more meat you put on your plate, the less plants, though, you can fit. So I personally, have meat once or twice a month. And I think that's probably a healthy way to do it. Have it as a treat and have really good quality. Mm-hmm. People want to cut, but most people in this country are having meat twice a day. Mm-hmm. So even if you went to, once a day, that would probably be a healthier switch. So I don't think we know the answer. We also, is it about the saturated fat in the meat? That's the problem. And choosing ones with less saturated fat, probably a good idea. Mm-hmm. But the evidence isn't that strong. But, some evidence that chicken is better than beef, and venison's better than, beef. Plenty of spare venison in Scotland, but no one seems to want to eat it. It's very strange.
It is very strange. I grew up on venison. I had a lot of venison in my childhood.
They can't give it away. It's really weird.
Our local gamekeeper would leave a deer on our doorstep, and my mum would gut it on the kitchen table. So I had quite an odd upbringing. I think that's interesting. What about all the meat eaters? What about all the carnivores out there? Because I know lots of people that are really thriving on carnivore diets and ketogenic diets with high protein. Have you had any analysis of their guts?
People who eat high amounts of meat, generally have poorer gut health than those that don't. People that can sustain a paleo diet or whatever for more than a few weeks are pretty rare. So there may be 1% of the population that might just have evolved to do that, but 99% of us can't do it. And these keto diets are incredibly hard to sustain. So and if the more you think about it, you're just sort of destroying the diversity in your gut health. So your immune system is going to go to pieces. You won't be able to get the fiber. So the only reason I see it is possibly for your, if you have an obese, pre-diabetic, diabetic patient, you want to make a switch, give them a keto program for a few weeks, as, some GPs are doing, just to get them off their meds and get them feeling good. But then you've got to transition them onto more of a gut-friendly diet. And I'm not a big fan of this sort of milkshake, these low calorie, keto shakes and things, because I think you need to train people in how to eat real food if you're going to sustain it for the rest of their life. But to get people off meds, I think, anything goes. So and if the studies show it works, then that's a good approach. So I think we should all be eating less meat in this country, basically. And fish isn't necessarily the panacea. The evidence that fish is really good for you is quite limited.
Okay, interesting. So we should all be eating a bit less meat. We should all be eating those 30 plus plants per week, ideally, and getting that variety in. And I'm a big fan of seasonal eating, and trying to keep with the seasons as well, although we are still reliant in the UK to get a lot of our foods from abroad.
The African season, yeah.
Yeah. So yeah, I think we can get things at their best if we eat more in season. But obviously I don't do quite enough gardening myself to make that so useful. What other tips do you have?
Well,
for
fermented foods, I think, are really underrated.
Yeah.
We've done a big survey we're going to publish soon, just showing, 50% of people, will see a benefit if they can go to 3 portions of fermented food a day. So I advise everyone listening, just go for it. You may just be having your low-fat yogurt or, and that you think that's it. Or, go in, do a couple of weeks trial, just having at least 3 portions of different fermented foods. And in our study, over 50% noticed significant benefits just of doing that. It's really good for the immune system, and that it reduces inflammation. And I think that's probably how it works mainly. So I think that's a really big feature. We've discussed time-restricted eating, switching many more beans, legumes, because they're not only high in protein, but they're also high in fiber. A lot of talk about us all being protein deficient. I think that's complete nonsense. 90% of us are absolutely fine, whereas 90% of us are deficient in fiber. So if any doubt, just get more beans and legumes, chickpeas on your plate, and that will sort out both. And really try and avoid ultra-processed foods that you don't know are there. There are lots of hidden ones for you and your kids that are quite hard to spot. Read the back of those labels, until we come up with a nice app to help you. And you don't have to cut it to 0 because that's virtually impossible. But levels are currently 60% in the UK of our food coming from UPF and 70% in children. I mean, that's the worst in the world. So anything we can do to reduce that is going to help your gut microbes.
It's frightening, isn't it? I mean, just the additives that are in our ultra-processed foods, like the emulsifiers and things, are really concerning. So yes, we want people to be really looking at the backs of packets and taking note of all of this stuff. Before we go, Tim, can you just, is there anything in microbiome research that you are really excited by at the moment? Anything that you think, watch this space, or, where we're really, something that is spiking your interest just now?
I'm really interested in the virome.
Mm-hmm.
We're talking about the little phage viruses that eat bacteria. Mm-hmm. And I think we're getting close to be able to measure them properly and see how we can manipulate them as possibly new treatments to change our gut health and have impacts in things like cancer and things. So I think it's a really exciting area. It could take 10 years, but I think, we're now getting snapshots of what's going on in our gut. It's a whole other dimension. I think that's really cool. Not for prime time yet, but it's just one of those, if you're looking at this big dimension of what's going on in our guts, I think that's really exciting. But there's so much, we need another hour to talk about it all, right?
We do. And maybe when you've researched a bit more into the virome, you can come back and talk to our listeners about it. Fantastic. Thank you so much, Tim. This has been really interesting for me, just to hear about your research, where you're going with it all. I think Zoe has been absolutely superb in terms of bringing microbiome research to the forefront, highlighting it to everybody. You know, the work that you've done with that is excellent. And I think without it, we would be, you know, way back in terms of our understanding of microbiomes and their importance for health. So thank you for all the work that you've done. And thank you for keeping going with all of this. And please do, when you've got some more of your Zoe research, do come back and talk to our listeners about it, because we would love to hear more updates from you about this really exciting field. Is there anything else you want to say before we head off?
Tune in also, you know, to our podcast for about all doctors need to know more about nutrition. That's true. And, you know, I learn a lot from them. So they're great. But I'll come back and fill you in on when we get some more discoveries.
Brilliant. Thanks so much, Tim. Thank you so much. Bye for now.
Bye.
Thank you so much for listening to this episode of Microbiome Medics podcast. We really hope you enjoyed the content and we welcome your feedback. We'd love to hear any suggestions you might have for microbiome topics that you'd like us to cover. And we also appreciate listeners' questions and will endeavour to answer them in the next podcast.